Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Justin Sonnenberg. Dr. Sonnenberg is Associate Professor of Microbiology and Immunology at the Stanford University School of Medicine. Dr. Sonnenberg's lab is interested in the basic principles that govern interactions within the intestinal microbiota and between the microbiota and the host. Today, Dr. Sonnenberg presents his session, Gut Microbiota Targeted Diets Modulate Human Immune Status. Dr. Sonnenberg, over to you.
It's a pleasure to be with you today. I'm here to talk to you today about our gut microbiome or gut microbiota, this complex community of microbes that lives in our gut. My lab studies how these microbes are influenced by diet and how the changes that can be induced by diet go on and impact our health. And to look at these questions, we use a variety of tools. We use a mouse model. We develop a lot of technology to study the gut microbiome. We also study traditional populations of humans, populations living traditional lifestyles like hunter-gatherers, shown here, the Hadza of Tanzania. And then more recently, we've actually begun doing dietary interventions in humans. And so I'll tell you a bit about all of these different approaches and the insight we're getting into diet, gut microbiome, and health. So I like to start off with this cross-section of a colon showing the gut microbiome in situ. You can see all of the microbes in the top left of this image, all of the little colored rods there. Each one is a different bacterial cell. And these bacterial cells are sitting up very close to our colonic tissue here in blue. You can see just separated by this thin green mucus layer that we secrete to keep the microbes in the right spot. You can immediately see how dense this community of microbes is and how close it is to our tissue. On the one hand, these microbes are an important component of our biology. On the other hand, we can even think about these microbes as this incredible interface for exacting change on our biology. We know that we harbor trillions of microbial cells. By cell number, we're actually more microbial than we are human. We know that there are incredible collective coding potential in these microbes. They have 100 times more genes in their microbial genome than we have in our human genome. And we don't know what most of these genes do. And the field is rapidly moving from not only understanding who is there, which microbes inhabit our body, but what are they doing? And while we have microbes all over our body, on our skin, in our nose, the urogenital tract, we know that most of these microbes are in our distal gut, in our colon, and that'll be the focus of my talk today. So we've known for a long time that our gut microbes actually are important in helping digestion, are important in gut health, but we've also come to realize that these microbes influence much of our biology outside of our GI tract. They influence our metabolism, our immune system, and can even impact neurodegeneration, moods, and behavior in animal models. So it seems that there is no part of our biology that's untouched by our gut microbes. And what is special about these microbes is that they're not just part of this integrated complex system that is our body, but they're also malleable. It's much easier to change our gut microbiome than to change our human genome. And because they're connected to so much of our biology and they're highly individualized, it's very likely that they will be an important part of precision health going forward. They will provide an important lever on our biology. We just have to learn the rules and understand how to manipulate these microbes in ways that benefit our health. And so a major question in the field is, to what extent can we manipulate our own biology via the microbiota? What tools and knowledge base do we have to develop to do this? We know that there are many approaches to manipulating our gut microbes, extending from fecal transplants to prebiotics to drug targeting of the gut microbiome. But we also know that diet is one of the dominant levers that we have for changing both our microbial composition and our microbiome function. And so, of course, these microbes are an important part of our biology, but there is an interesting asymmetric plasticity of our gut microbes and our human genome over evolutionary time. We know that our gut microbes can change very rapidly, where our human genome evolves relatively slowly. And so it leads to this potential problem where if we exact lifestyle changes and introduce medicines that can negatively impact our gut microbiome, the question arises, can we change it to a point where it is no longer compatible with our human genome? Could there be an incompatibility between our human genome and our microbiome brought on by lifestyle, dietary, medical changes? So we're very interested in this idea that we may have somewhat of a foreign entity living inside of us in the industrialized world that is misregulating aspects of our biology. Now, important background here is to set up the logic of what components of diet play a disproportionate role in impacting the gut microbiome. This slide just shows a schematic of the small intestine and large intestine. Most of the microbiota lives in the colon, in the far reaches of the digestive tract. And so that means if you're eating a Western diet, shown in the bottom in brown, the simple nutrients, the simple carbs, fats, proteins in that Western diet get rapidly digested and absorbed in the small intestine, leaving very little for your gut microbes. If you're eating a plant-based high fiber diet, which is rich in microbiota accessible carbohydrates, or MACs, those complex carbohydrates are not digestible in our small intestine and make it to the distal gut, where they serve as the primary fuel source for gut microbes. And we know when gut microbes ferment complex carbohydrates, they produce beneficial metabolites like short chain fatty acids that get absorbed into our bloodstream. So if you're eating a Western simple carb diet, your blood chemistry looks very different than if you're eating a high fiber diet. And so one thing that has become evident in the field over the past decade or so is that the industrialized microbiome appears to be losing diversity. And even in the context of industrialized populations, higher diversity appears to represent better health. And so it leads to this concern that various aspects of our industrialized lifestyle, like our Western diet, sanitization, certainly important during a pandemic, but potentially having negative impact on our microbiome, antibiotics, baby formula C-sections, and various other lifestyle changes are leading to biodiversity loss that's having a negative impact on our health. And so a former student of mine, Sam Smits, was interested in how the microbiome has changed globally based on lifestyle. So he brought together all of the microbiome data that was available at the time that he compiled this graph for different populations around the world. And he put all of this microbiome compositional data onto one graph, and that's shown here at the top. Each bar represents a different population's gut microbiome microbial composition, and separated along an axis that differentiates these samples by the largest amount of variation that can be detected in the data. And so here we see a very stark contrast. All of the populations that live traditional lifestyles move to the left of this axis, and all of the industrialized populations move to the right of this axis, showing that lifestyle, independent of where you live on the earth, really greatly impacts the gut microbiome. And we also know that this industrialized shift in the microbiome is coincident with a loss of biodiversity. So moving from this complex, diverse ecosystem to a more simplified, less diverse ecosystem. Now that cross-sectional data matches nicely with another beautiful study performed by Dan Knight's group at the University of Minnesota, where they tracked individuals that were immigrating to the United States from Southeast Asia. And as these people moved to the United States, they saw in real time over the course of months, westernization or industrialization of their gut microbiome, loss of Prevotella strains, loss of fiber degrading enzymes in their gut microbiome, and increase of Bacteroides. Within just 9 months, they saw these changes, and the graph on the right shows that the longer these immigrants are in the United States, the more profound the changes in loss of here Faith's PD, which is a measure of diversity. So diversity goes down the longer that immigrants are in the United States. And so there are a lot of factors that correspond to lifestyle that could be impacting the gut microbiome. We think diet is a really important factor, but as I mentioned, there are a lot of other factors that are likely playing a role. We were curious how important diet was if taken in isolation in impacting gut microbiome diversity. So we used a mouse model. We gave mice a human microbiota and gave them a high MAC or high fiber diet. Remember, MAC stands for microbiota accessible carbohydrate. After the microbiota stabilized, we broke this group of mice into 2 different arms. Group 1 continued to receive the high fiber diet, high MAC diet. Group 2 received the low MAC diet. And we were curious how this would impact diversity and whether if you reintroduced a high MAC diet, could you get a low diversity microbiota that resulted from the low MAC diet to recover? So this is basically asking the question, is it too late for us, or can we start to eat a healthy diet and help our microbiome recover? We actually performed this experiment over multiple generations. So we took mice and put them on either a high fiber or a low fiber diet over the course of 4 generations. And so on the high fiber or high MAC diet over 4 generations, this is the data. So this is the persistence of strains over the course of those 4 generations. In this graph, each column represents a different bacterial species, and the different rows represent different mice in different generations, generations 1, 2, 3, and 4. And the colors represent how abundant that microbe is in that mouse. So we see on the far right here, there are many microbes that are very stable over the course of the 4 generations, other groups that are more variable but still maintained, and then a few on the far left here that are lost over the course of the 4 generations. So some loss of biodiversity on the high fiber diet, but what does the low fiber or low MAC diet look like? Well, we see this massive extinction that occurs on the low MAC diet, and that loss of species compounds over the course of the 4 generations. So here we see that elimination of dietary fiber depletes the gut microbiome irreversibly. And importantly, in the 4th generation mice, we put the mice back on a high fiber diet, and we see that even though they're eating a high fiber diet, they're not able to recover the species that they've lost. So once species have been lost from the ecosystem, it's difficult for them to recover just based on diet. So a major question is, how does an industrialized microbiota impact our health? Well, we know that there are many inflammatory and autoimmune diseases that are on the rise around the world. So we know that ulcerative colitis rates are increasing, many forms of inflammatory bowel disease are increasing. Other inflammatory disorders like autoimmune diseases are on the rise. And we know that many of these diseases are driven by chronic inflammation. And we also know that the gut microbiome is a critical modulator of our immune status. So it leads to this hypothesis that our deteriorating or changing gut microbiome is changing our immune system. And that different immune set point is leading many of us to an increased probability of having an inflammatory disease in our lifetime. So a major question is, can we repair our gut microbiota and improve our health? And we were very interested in this question, and specifically whether we can change our immune status, that is, decrease inflammation, with diet-induced microbiome alterations. And so we started the Center for Human Microbiome Studies at Stanford. This is a center that its main purpose is to conduct human microbiome studies using dietary interventions. And we have a great group of partners here. And the whole point of this center is to basically reverse the translational pipeline. Typically, in biomedicine, we work from molecular mechanism through animal models up to humans, hoping that we can bridge this large translational gap. The point of this center is to reverse this and basically start with dietary experiments in humans, and then reverse translate into animal models to understand molecular mechanisms. And so our first study in this area we call the Phi-Pho study, fermented fiber food study. And the big question here is, can we modulate the immune system via a microbiota targeted diet? We picked two diets that we know have a large chance of changing the gut microbiome, one in which we introduce a large amount of dietary fiber using plant-based food, and another arm where we introduce a high fermented food diet based on common fermented foods. This is a wonderful collaboration with Christopher Gardner's group. They perform the dietary interventions. They enroll people. We take blood and stool samples at baseline during a ramp period, during a 6-week maintenance period, and then we give participants their choice of how they want to eat for a washout period. And in the end, we perform microbiome profiling on the stool and immune profiling on the blood samples to get a broad view of how the immune system changes over the course of these dietary interventions. This is a project that's been driven by a wonderful group of trainees, Maddie Toth, Hannah Wasteek, and Gabby Fragidakis. And this paper was recently published in Cell in August, if you're curious about reading more of the details. So first, focusing on the fermented food diet, people ate diverse fermented foods, but they ate very differently. We encourage people to go with the fermented foods that were comfortable for them. We asked them all to eat as wide a variety as possible and as much as possible over the course of a day. And these are the number of servings. Each graph represents a different individual, and this shows the different fermented foods that they eat, number of servings over the course of the study. So the week's time is the x-axis. You can see some people ate a lot of fermented vegetables. Other people drank a lot of kombucha. People achieved their fermented food consumption in different ways. The only requirement was that people had to consume a fermented food that contained live microbes, and those live microbes were involved in, of course, fermenting that food. So this is a graph showing the fermented food servings intake per day across the fermented foods group. They started off at baseline consuming almost no fermented foods and got up to over 6 servings of fermented foods per day on average during the maintenance phase. And we were curious what happened to their gut microbiome over this intervention. We see that gut microbiome diversity increases over the course of the intervention. So exactly the change that we would hope to see with a healthy food-based intervention, increasing gut microbiome diversity. And so we were curious in this gut microbiome diversity that we observed whether those bacteria were coming from the fermented foods themselves or from somewhere else. And so we actually brought a lot of the fermented foods that these participants were eating into the lab. We cultured microbes, identified microbes using sequencing methods, and then asked the question of whether those microbes are found in the gut of these individuals that are increasing their gut microbiome diversity. And what we see is the vast majority of new microbes in their guts are not from the food. Only about 5% of the new microbes are from the fermented food. The vast majority of diversity is from somewhere else. And so we don't know if these microbes are coming in from elsewhere and engrafting in the microbiome, or if they were below the level of detection and are blooming in these participants during the fermented food intervention. But it appears that the fermented foods are catalyzing a gut microbiome diversity increase, acquiring microbes from somewhere else than the fermented foods. So a major question is what happened to the immune system as these people consumed fermented foods and increased their gut microbiome diversity? We see the markers in their serum, there was a broad decrease in inflammatory markers. So each one of these graphs shows a different inflammatory marker and its levels in the participants relative to baseline on the far left. We can see that all of these markers, including IL-6, IL-18, IL-12, these are different cytokines that are associated with inflammation, all decrease over the course of the dietary intervention. And of course, it wasn't just these 6. We actually saw close to 20 markers that decreased over the course of the dietary intervention. This decrease in inflammation was not confined to cytokines and chemokines. Performing mass cytometry or CyTOF on immune cells that were collected in the blood, we also see that many of those cells show a decrease in phosphorylation of signaling cascades that are associated with inflammation. And those phosphoproteins are associated with different cell types. So we see this broad decrease in inflammatory cell signaling also occurring over the course of the dietary intervention. So it appears that fermented foods increase gut microbiome diversity and decrease inflammation in this cohort of 18 healthy adults that we were looking at. Now we're curious in how the fermented foods are mediating these changes in the immune system. And so we have developed a pipeline for surveying the metabolites that are present in different samples. This metabolomics pipeline is focused on gut microbe metabolites. And so now we're surveying serum of these individuals in these trials to determine if there are specific chemicals that the microbiota secretes into the blood that are correlated with different immune markers. And this gives us molecular candidates that we can then bring into the lab and begin to test in cell-based assays for molecular mechanisms. So some of that reverse translation that I mentioned earlier. Now, the high fiber diet was a high fiber food diet, and that was focused on getting people to eat a lot of whole grains, legumes, vegetables, fruits, nuts, and so forth. And so we see, similar to the fermented foods, that each individual accomplished high fiber in a different way and eating a diversity of different food groups of high fiber foods. So each one of these graphs shows a different participant, and we can see that some individuals ate a lot of grains, other individuals ate a lot of fruits, but they all were able to achieve a high fiber diet using a diversity of high fiber foods. Now, we see that monitoring dietary fiber at baseline, these individuals were consuming around 20 grams of dietary fiber per day. During the maintenance period, they got on average to over 40 grams of dietary fiber per day. So that's doubling their fiber consumption. Yet despite that large increase in fiber consumption, we did not see an increase in microbiota diversity. That's the graph shown at the bottom. The microbiota diversity stayed relatively flat over the course of this intervention. And just to remind you, that contrasts with the fermented food group that increased their gut microbiome diversity. So the major question is why? We're giving these individuals a high fiber diet. Why are the microbes not becoming more diverse? Why are they not recruiting new microbes? And we think that one hypothesis here, that the industrialized microbiota may not be able to degrade many complex dietary carbohydrates. The diversity is lacking, and these fibers are passing through these individuals untransformed. And we actually surveyed the stool for carbohydrates of these different participants. And we see that indeed the amount of fiber in the diet is highly correlated with the percent of carbohydrate that's present in the stool. We see many of the monosaccharide markers of hemicellulose and pectin that we would expect to be fermented coming out in stool, an indication that the microbiota of these individuals is not doing a very good job fermenting that dietary fiber. And to connect back to the immigration study that I mentioned before, another facet of their study was looking at fiber degradation capacity. And what they see is a great loss of the enzymatic capability of the gut microbiome to degrade complex carbohydrates. So it does appear that the industrialized microbiome is lacking much of the ability to degrade the complex carbohydrates found in dietary fiber. And to remind you of our multi-generation experiment, as we put the 4th generation mice back on a high fiber diet, those mice were unable to regain all of the diversity that had been lost in the microbiome. We also performed the experiment of fecal transplant, taking the microbiome of the high MAC diet that has all this diversity and doing a fecal transplant to provide those microbes to this depleted gut microbiome of the low MAC fed mice. And so that data is shown here. We see the generation 4 mice with a low diversity microbiome getting the microbiome from the FMT donor mice, the 4th generation mice fed the high fiber diet. And we see once they get the FMT, within 3 days, they recover all of that diversity and it persists day 6, day 10, day 14. All of that diversity persists in the gut microbiome as long as those mice are fed a high fiber diet and given access to acquiring those microbes. The control mice that were given a sham FMT, not containing microbes, were unable to acquire that diversity. So it appears that diet-induced extinctions in the gut microbiota compound over generations, and that providing those microbes back along with the high fiber diet can restore diversity. This is a perspective that I wrote with my wife, Erica Sonnenberg, who runs the lab with me at Stanford. And we propose this depleted gut microbiome that's occurred over the course of recent human dietary change may require not only dietary change, but also species reintroduction to reacquire important diversity that we've lost. And I just want to finish by thanking all the members of my wonderful laboratory, my wife, Erica, and thanks to all of the funding agencies that have supported us. Thanks to many of the donors out there that have supported our dietary intervention studies and other work in our laboratory. And then also mention that my wife and I have written a book called The Good Gut to help non-scientists or non-microbiome specialists understand the gut microbiome better and to understand how the gut microbiome is important in our health and we can impact its health through decisions that we make in our daily lives, such as what we're eating. And thanks for the invitation to speak.